Dr. Madangi Ganapati has obtained her MSc in Genetics and Plant Breeding from Kerala University and PhD from School of Biotechnology, Madurai Kamaraj University. Having a passion for gaining knowledge and for instilling curiosity in fertile minds, she has been active in the field of teaching and research for more than 15 years. Her current research interest includes recombinant protein expression in plants, transgenic plant development and manipulation using molecular techniques, and analysis of changes in basic gene regulatory pathways during transgene expressions, nutraceuticals, and the role in alleviating certain chronic human diseases. She has a number of publications in peer-reviewed international and national journals.
Welcome to the lecture series on food microbiology. In today's session, I'll be talking about 1. The viruses, 2. How do we classify the different microbes, what are the different classifications of microbes, bacteria which are present in the food, and then the general classification is based on gram-negative, gram-positive, etc. The specific classification would be based on what are the metabolites or what are the type of nutrients which they utilize. So accordingly, the type of food which they normally prefer. So based on that, we will be talking about how these organisms can be classified. Now, the first are going to be the viruses. Foodborne viruses, for example, hepatitis A, Norwalk-like virus, etc., which can cause gastrointestinal diseases. So this is a negative aspect. But some viruses can be used to identify a particular foodborne bacterial pathogen like Salmonella, Staphylococcus aureus strains, etc. So what do we do is, remember, a virus inhabits only a living cell. So suppose you suspect that a particular food has been contaminated with a particular, for example, Salmonella. You, instead of trying to identify, so what do? You take a swab or scrape a piece of the particular food, grow it in the nutrient medium. You need to know. So you can count what we call as the bacterial colonies growing up. So what do is, now you need to identify what is the bacteria growing there. So for that, suppose you are going to suspect that it is Salmonella, you have a specific bacteriophage which normally infects Salmonella and causes lysis. So to this particular growth medium where you are growing this organism, introduce the bacteriophage. So what will happen? This bacteriophage will come and infect the Salmonella. Then after a particular time, the virions will be released, causing a lytic cycle. The bacterial cell will lyse. So this will be counted as the bacterial plaque. So for the identification, so once you know that what is the sort of bacteriophage which you have used, and you also know that this particular bacteriophage normally infects only this bacteria, you can identify that yes, the given food sample contains this particular pathogen. So for that, we can use bacteriophages. Then the next aspect is the bacteriophages can be used to transmit specific genetic traits to bacteria. That is, suppose you want to improve a particular strain. You already are using a particular strain in a particular industry to produce a particular flavor or a nutrient, but you want to improve, and you are taking the gene which you want from some other strain. So how to deliver it? For that, the bacteriophages can be used as a vehicle. So this is normally done in the case of Lactococcus lactis. The negative aspect is these viruses or the bacteriophages can cause fermentation failure, especially in lactic acid bacteria. As I said, lactic acid bacteria are the ones which are used as starter cultures for yogurt, cheese, for so many things. So if these lactic acid bacteria lyse, then fermentation will not happen. We call it as fermentation failure. In such a case, you don't want the bacteriophage to infect. You want the culture to be pure. You don't want it to be infected with these viruses. So that is also the negative aspect. Now, the classification of bacteria based on their nature. Here I am going to talk about only those particular bacteria which are more important to the food industry. As we saw in the last class, gram staining is an important means to identify whether an organism is either gram positive or gram negative. All of you know that this positive or negative is based on the thickness of the peptidoglycan layer. So here you have certain gram negative aerobes like Campylobacter, Pseudomonas, Xanthomonas, etc., which are normal food which do cause spoilage on food. Then you have the gram negative facultative anaerobes, Escherichia, Enterobacter, Erwinia, Salmonella, Shigella, etc. Now, Escherichia is a common human gut microbiota, is normally present in the human gut microbiome. So this is actually an opportunistic pathogen. Normally it is friendly, but if an opportunity comes, it can become a pathogen. Whereas the other gram-negative facultative anaerobes like the Enterobacter, Erwinia, Salmonella, Shigella, all are pathogens. They cause various gastrointestinal diseases. Then among the gram-positive cocci, example Staphylococcus, Streptococcus, Lactococcus, Enterococcus. Again, the Lactococcus normally is not a pathogen, we use it in the food industry. Staphylococcus and Streptococcus are normally present on the human beings, on the skin surface, etc. But they can also be opportunistic pathogens. Enterococcus is normally a pathogen. So, gram-negative aerobes you have, gram-negative facultative anaerobes you have, gram-positive cocci you have. The next class we are talking about is gram-positive endospore-forming rods. Now, this is a major aspect which causes foodborne disease, very lethal. Endospore, as you know, are formed by a certain group of bacteria when the conditions are not conducive for normal bacterial growth. So, these endosperms which are formed during the non-conducive environment are very hardy structures which can withstand very high temperature, very high pressure. They are dormant structures. The metabolism of the bacteria goes down to almost standstill inside the endospore, and this endospore can remain in the environment or in the food for eons together without getting destroyed. And when the situation again becomes conducive, the endospore will germinate, giving rise to the bacteria. A typical example is Bacillus, which is a gram-positive spore-forming bacterium. Then you have the Clostridium species again, which also forms an endospore. Now, in the case of Bacillus, the Bacillus cereus, subtilis, etc. are to some extent beneficial to the food industry because they produce extracellular enzymes that can hydrolyze the carbohydrate, protein and lipids. Now, this can be utilized by the food industry, but you do have Clostridium which can form spore. Now, Clostridium botulinum, for example, is a typical example which causes food poisoning. It is extremely lethal. This botulinum spore which is formed can survive in the environment, and spores, as I said, are hardy structures. Normally, with what we talk about destruction of microbes, autoclave, or you are exposing the organism or the bacterium or the foodstuff to 121 Kelvin, 121 psi pressure at high temperature, you retain the organism within that, the organism will be killed. And you require that 15 minutes of the retention to kill the spore. But your normal high temperature or normal just high pressure or any chemicals normally will not destroy these spores. So, in such a case, if the bacterial spores are present in the food, then your food processing mechanism would have to be even more specific to kill the spores, especially if the Clostridium botulinum spore is present, because this causes death to the person consuming it. If the spore has been identified, or if the food contains the microbes, if you suspect that the food has this gram-positive microbes which can form spores, then not only the microbes but then their spores also will have to be destroyed during the preservation and processing technology. Among the gram-positive, you have non-spore forming organisms also. Then Corynebacterium. Corynebacterium glutamicum is normally used in the food industry to produce glutamic acid. Like that, you have the Propionibacterium, which is used for the industrial production of proline, for the production of propionic acid. This is also used for the production of Swiss cheese in cheese manufacturing industry, especially for the production of Swiss cheese. This Propionibacterium is used. Now, the other classification is based on similarities, meaning one, the type of nutrients which the organisms, this group of organisms will use these nutrients based on that, or the other aspect would be based on the metabolic pathways. If I have this nutrient, this would be the metabolic pathway would be followed, this would be the end product. So, based on such similarities, we can again classify bacteria. The typical group 1 consists of lactic acid bacteria. So here, this lactic acid bacteria, the group contains microbes which produce large quantities of lactic acid from carbohydrate. So carbohydrate is the source, this carbohydrate is broken down, the end product is lactic acid. Example for this would be leuconostoc, lactococcus, etc. The second category is acetic acid bacteria. Example is Azotobacter azeti. As the name indicates, acetic acid would be the end product. Whatever be the source, whatever be the nutrient source or the energy source, the end product is going to be acetic acid. Then propionic acid bacteria. These are mainly used in dairy fermentation. As the name indicates, the end product is going to be propionic acid. Like that, we have the butyric acid bacteria. Then we have the proteolytic bacteria. Now, the proteolytic bacteria, as the name indicates, degrade proteins. So for the degradation of these proteins, they synthesize extracellular enzymes. Bacillus, Staphylococcus are examples. So the two aspects here is, one, utilize these for breaking down protein. Two, identify the extracellular enzymes. Then you can have an industrial production of these enzymes and market the enzymes. So there are two aspects in the case of utilizing the proteolytic bacteria. Like that, you have the lipolytic bacteria. As the name indicates, these hydrolyze triglycerides with the help of extracellular lipases. So triglycerides can be broken down, the food can be made more easily biodegradable because they have already been digested by the lipase. The second aspect is you identify what is the type of extracellular lipase which the organism synthesizes, and then find out a mechanism to purify it from the organism. You can market it. That is the second aspect. Then you have the saccharolytic bacteria which hydrolyze complex carbohydrates. Typical example is the Bacillus, Enterobacter, etc. The next category we are talking about is thermophilic bacteria. Thermophile, as the name indicates, they can grow above 50 degrees centigrade. Bacillus, Streptococcus, Lactobacillus, etc. are examples, which means that if the food has been processed at less than 50 degrees centigrade, you would not have destroyed the bacteria present, because they can survive even at temperatures beyond 50 degrees. So in such a case, if the food has to become free from these microbes, then the processing temperature would have to be much higher, because they can survive in temperatures. So you will have to go to say 80 degrees or 90 degrees, find out the upper threshold level, and then process the food at that temperature so that these bacterial species would be completely destroyed in the food. You have the psychotrophic bacteria. These grow at low temperatures, less than or equal to 5 degrees, which means that the normal refrigeration, what you have in the refrigerator, these organisms can grow there. So the vegetables and the fruits which are stored inside the fridge, they can have infection of the Listeria and Yersinia, because these can survive. Normal bacteria which survive in the temperature of 20 to 37 will not survive inside the fridge, but these organisms which can grow in temperature less than 5 degrees can grow. So those type of food can still get destroyed in spite of being refrigerated. Then you have the thermoduric organisms. These can sustain even the pasteurization temperatures. So you mean that your processing cannot be pasteurization. You would have to have some other mechanism to preserve such type of food, to help to prevent it from being inhabited by Micrococcus, Enterococcus, etc., which are thermoduric. Halotolerant, as the name indicates, these organisms which are halotolerant can survive very high salt concentration, which is greater than 10%. So your pickling will not help. In spite of the high salt present, the organisms can still grow, because what is growing up can grow in that high alkalinity. Typical example is Vibrio corynebacterium. Likewise, aciduric. These survive in low pH, less than 4. So to kill these organisms, either you will have to raise the pH or devise some other mechanism, because low pH alone is not sufficient to kill these aciduric organisms. Osmophilic. This indicates that the organisms can grow in higher osmotic environment. So your processing and preservation mechanisms would have to take this character into consideration, and you will have to devise a novel strategy to overcome the growth of these osmophilic organisms in the prepared food product. Then you have the gas producing bacteria. These gas producing bacteria give out carbon dioxide, hydrogen sulfide, etc. during metabolizing the nutrients. This evolution of carbon dioxide, we already saw how it is utilized in the bread industry, because that is what gives the spongy texture. But as I also said there, the bread will collapse if there is too much of fermentation and high excess of carbon dioxide production. Now, the evolution of hydrogen and hydrogen sulfide, for example, when the proteins get broken down, you get, when the bacteria degrade the proteins, hydrogen sulfide is emitted. The typical smell of rotten eggs, as you know. So this indicates that the food has been destroyed, it is degraded, it is not fit to be consumed by human beings. So this also will indicate the presence of Leuconostoc, E. coli, etc. So the evolution of the gas is normally taken as an indicator for the presence of certain microbes which are present in the particular foodstuff. You have slime producers. The slime producers synthesize complex polysaccharides. Example, Xanthomonas enterobacter. This slime would be deposited on the outer surface, changing the texture of the food concerned. That nature itself will indicate that the food has been spoiled. So that is the major classification. Then you classify them based on as spore formers, for example, Bacillus, Clostridium, aerobes. Here the growth of aerobes can be controlled in the food if you change the atmosphere, how you pack, how you process, because they do require oxygen. Anaerobes will have to be controlled only when you give oxygen, because these anaerobes will not be able to survive when higher oxygen is present. So you modify the atmosphere in the case of aerobes and anaerobes to prevent their growth in foodstuff. Likewise, facultative anaerobes, these do have a tolerance level. Even though they are facultative, the amount of oxygen carbon dioxide concentration in the atmosphere in the food, where the food is being processed and preserved, can be modified to prevent their growth. Then the last is the coliforms, fecal and the enteric pathogens. The general class coliforms indicates E. coli, Enterobacter, Klebsiella. Now, as I said, E. coli is an opportunistic pathogen. Enterobacter and Klebsiella are normally present in the food. So you have certain identification methodologies which will tell you whether the coliforms are present in the food. The second question is, if the coliform is present, what is the amount? You have a threshold value which is accepted. If the presence of the microbe is beyond the threshold value, then the food has to be rejected, it is not fit for consumption. The fecal coliforms, these are normally those organisms which indicate that sewage has been mixed with drinking water. This is what we utilize for testing the potability of drinking water. If fecal coliforms are detected in drinking water, it means that there is a contamination somewhere, that water is not good for drinking at all. So this indicates the level of sanitation which is present in the particular area. The last class is the enteric pathogens which cause diseases. How do these microbes come into the food? That is the next major question. Now, your food can be plant products, it can be animal, bird, fish or shellfish, any animal product. Now, in the plant product, where does this microbe come from? Now, the plant grows, right? You sow the seed in the soil, it grows up to form the plant. Now, the soil or the rhizosphere is a big repository of microbes. So, this can be taken up. You have the fungal spores, you have some organisms, you have the nematodes which are present in the soil, then you have the microbes, for example, which can get through the root rhizosphere into the plant. Then on the outer surface, as the stem grows up, the fungal spores are present in the atmosphere, the bacteria are present in the atmosphere that can infect the plant. So in the various stages of the growth, the microbes can infect the plant. Now, when you are eating a plant product, if the plant had been infected by the microbe during its growth phase, the remnants of that, either the bacteria or the fungal spores or the metabolites of the particular organism, would be present in the food. And if that food is being consumed raw or the processing is not proper, then the human being who consumes this particular plant product would also get infected. So that is one aspect. Animals. Here we talk about how the animals are housed. What is the source of the meat? Are you going to have meat from the stray cattle, or are you having a farm where the cattle is grown? Then here we call about then what is the hygiene practice present on the particular farm. For example, the animal can be infected by enteric organisms, microbial infection, for which it will take the veterinarian will supply antibiotics. That antibiotic remnant or the residue would be there on the meat of the infected animal or in the food you take. It will be even present on the milk if you are taking about a dairy farm. The second aspect is on the outer side, the skin of the animals. They do contain hair or the hide. There also you have the microbial dwelling. So when you remove the hide or the hair and process the meat, if sanitation is not complete during processing, the microbes will get into the meat which is being processed. So there again you have a second chance of contamination. Like that in the case of birds, the skin, the feathers on the surface of the fish, shellfish, here these contain certain toxins in the marine environment. It can be because they feed on the algae, or then again process it. We have the dinoflagellates which can attack them, giving rise to toxins which are present in the shellfish. So when the human beings consume them or the food is in profit, you will get poisoning. So plant and the other animal, bird, fish or the shellfish products do have chances of harboring microbes. So you will have for each of the processing step, whether it is home-based processing or whether you are talking from the point of view of an industry, precautions will have to be taken to avoid the growth of the microbes. It can be spoilage, it can be poisoning, it can be intoxicants. In either way, you will have to have a prevention methodology for the growth of the microbes or the associated toxins in the particular food concerned. Then air, the atmosphere, as you know, has humorous fungal spores. It is a rich repository of microbes. So if you are having an open space where you are processing, naturally when we are cutting open, for example, you are chopping, then your meat, for example, that once the external hide of the skin of the animal is actually a protective layer. You remove the skin, you have the rich meat, and it is exposed to the atmosphere. So atmosphere has the microbes which in turn can use this meat as a repository. So here you already have an infection. So if your processing is not complete, it is this microbe infested food which you are going to consume. So air, then soil. Do you have soil contaminants present there in that particular area where your food is being processed? So that can also be a source. Then sewage. Now this comes, we don't necessarily go and use sewage water to clean, but if the water which you are going to use for processing the food has a contamination with sewage, then your sanitation is not proper. The microbes will be associated with the food because of that. So these two are associated properties, sewage and water, they can become mixed. So the quality of water which is used for processing as well as for cleaning. So these two become very important as the sources of microbes in the food. Humans. Now, when we are doing the processing, are you ill? Are you sick? Do you have microbial contamination? So then it is better that you keep away. So that you can become a source because your skin, your hair, your nails, hand, all are repositories for microbes which can be passed on from the human beings to the foodstuff concerned. Food ingredients. This is a major aspect because for example spices, we use pepper powder, we use clove powder, so many things as a flavors, additives. These contain fungal spores. So and remember, spores cannot be destroyed easily. So when you add these spores to the food, along with that food ingredients, you are adding microbial contamination to the food. So for this, during processing and preservation, we will have to take steps to prevent that. Equipment. Sanitation of equipments becomes very important, starting from the knives to the cutting boards to the huge industrial size equipments. Every step there has to be a periodical checking to find out whether you have microbial growth, and then sufficient steps have to be taken to prevent the growth of that and sanitize these. So this becomes very important. The last is miscellaneous, where whatever has not been included in all this has to be taken into step to prevent microbial infection as food. So in this particular episode, we talked about what are the different types of bacteria which inhabit different types of food. 2, depending upon what is the end product. For example, lactic acid bacteria produce lactic acid. So depending upon the end product, how do we classify the organisms? Then when we are talking about a food, what are the probable source from which microbial contamination can happen to the food, and how do we remove them? What are the steps we take to prevent? This particular lecture series will be continued later on with other topics. Thank you.